Listen, guys, whether it's my crowd, my tribe, or Dr. Miller's followers, I'm pinching myself because I'm so honored. You know, you know me. I'm a teacher. I'm a teacher of health. I diagnose cancer cells and then try to teach healthy cells. Also, I'm a huge NRF2 junkie. I've been introduced to NRF2 and then NAD and mitochondria since 2012. Been to 3 out of 5 world conferences, presented at 2 of them. So today, what are we doing? We're talking about cellular tapestry. I consider myself a Dr. Miller's groupie. I actually, in real life, I haven't met the guy, but I have been, sort of a client of following functional, what is this, functional genetic analysis?
Yes.
Functional genetic analysis probably since 2016, I would say, and absolutely accepted every Bob's webinar. So I started listening to him in US every time I get all the invites. And as of late, it was amazing because he was presenting more. And I was absolutely mesmerized by his explanation of cellular crosstalk. That was absolutely unbelievable because, guys, for me, we're talking either cellular symphony, how the cell interacts, or we're talking cellular cacophony. But we need to know that. And I think there's nobody better. I've sent you, I Facebook Dr. Miller's credentials. He's absolutely incredible. He is on, despite that he calls himself conventional naturopathic doctor. This guy is on forefront of medicine. He is building epigenetics. He is building enzymatics. He is building proteomics. He is ultimately talking nutrigenomics because to every cellular biochemical pathway or target or protein complex, what are we touching in health? Hopefully we're not touching the drugs, but we're touching health. We're touching foods. We're touching behavioral patterns. We're touching something that we can control to create this cellular tapestry in synchrony and symphony. So thank you. Today we're going to be talking my favorite things as NRF2, NAD, NADP, NADPH. Guess what? Glutathione. And obviously the king or queen of the cell, which is mitochondria. I call it the mother of the cell of mitochondria. Thank you, Dr. Miller. It's all yours.
Okay. Well, thank you so much. Thank you for those kind words. And as we said, I've not been medically trained. I'm just a traditional naturopath. But one of the things that people have observed is possibly because of my first career being in electronics, I looked at things just like an electrician would, and we create schematics. And so I think that's allowed us to look at things in such a new way. Let me first start out by saying that I think it's now very clear that the majority of conditions that we see in illness are related to inflammation. And what's interesting is that, the old-time naturopaths a long time ago that were considered quacks that, should be put in jail, were the ones who said that, we believe inflammation is the root cause of most of our problems. And I'll never forget about, 8-10 years ago, I saw a scientific article published that said, new discovery as to the cause of all illness. And I thought, well, this is interesting. And the discovery was that it's inflammation. So, that's something that we've been looking at for a long time. So having an electronic background and thinking schematically, I started looking at, well, if inflammation is the root cause, what causes that? And it really becomes quite simple. So what I started looking at is what are some of the factors that could create excess free radicals? And then we started looking at, well, what are some of the pathways that make antioxidants? Then we started looking at, well, what helps recycle our antioxidants? And what helps repair the cells? And what helps detox the poisons from the cells? So we took when you really think about what we do, it's ridiculously simple. We look at what are some pathways that might make more inflammation. Might there be some impediments to our ability to make antioxidants to neutralize that? And then how do we repair the cells and take out the toxins? And then in the traditional naturopathic philosophy, take care of that and just get out of the way, and the body's innate ability to heal kicks in. So that's essentially what we do. And it was about, maybe 12 years ago that we started learning about, through like things like 23andMe, that we could download the genome. And I think that revolutionized how we did things. And of course, back at that time, I had no idea what we were doing, but I thought, well, let's learn. So it's been a somewhat of a love affair since then. And so I spend about 16, 17 hours a day, either looking at genomes, researching. I have numerous people who help. We created the Nutrigenic Research Institute, and I have a whole team of people now who contribute. And then what we did is we took that original 23andMe data, and in the old days, we actually took an Excel spreadsheet and loaded it in, and did sort and all those kind of things, and started looking at some of the pathways. And now it's evolved into having our own genetic chip called Your Genomic Resource in our own online software that I've now spent about $2.8 million on to develop. It's an online platform that allows physicians, we only work with doctors, this is not one of these online things for the public, where physicians can download the data and analyze it. So it's definitely for the functional practitioner who's very serious about what we want to do. So if I can, maybe I'll start out with the concept of the pyramid. That might be a good place to start. So I'm going to do a screen share. And so I'm going to do a screen share of a pyramid here. And what this is, let me just do a screen share right here. Screen share number 1. There we are. And I believe you see the screen now. Okay.
Oh, we do. Awesome.
Yes. So what we talk about here, this is the pyramid. And so what we do, this bottom line, okay, each of these blocks is a set of genetic markers that, if there's a lot of mutations, has the potential. And when you talk about functional genomics, you all have to talk about potential, because this is not a diagnosis. A geneticist would look at genes that are related to disease. We're not doing that. We're just looking at genes that are related to creating the potential for less than optimal function. And that's why we call it functional genomics. So on this bottom line here, and I'll do some drawing, we look at, for example, if there's anything that would create the Fenton reaction. And we'll get into that a little bit. That's when iron combines with hydrogen peroxide to make what is called hydroxyl radicals. Again, a source of inflammation. Then we look at something called NOS uncoupling. And NOS uncoupling, I'm sure as everyone knows, nitric oxide is a very important molecule. We need nitric oxide to dilate our blood vessels, but it's also very much involved in reducing mast cells, the NOx enzyme. Then glutamate. Glutamate is what makes us intelligent, highly motivated, go-getter kind of person. But it needs to be turned into GABA. So glutamate's the intelligent, highly motivated, get-her-done kind of person. GABA's the don't worry, relax, be happy. If we have too much glutamate or not enough GABA, we have inflammation. Then there's all kinds of genomics that can be related to glutamate sensitivity. To gluten sensitivity, or to dairy, or to histamine. And there's many genes that make the enzyme called dynamine oxidase. And if we don't have that, we get inflammation. And then if we have time, we'll touch on a little bit on the heme cycle. And this is where, from the mitochondria, we take succinyl-CoA, combine that with glycine, and go through 8 steps to make our heme. Now, we tend to think of heme as hemoglobin, but heme is also involved with many processes of detoxification, and we'll show the map on that. And then one of the things that we're talking about is mast cells. Now, I'll have to admit, when I did traditional naturopathic training, 25 years ago, we didn't even talk about mast cells. And now it just seems as though so many people are talking about mast cell activation, where these mast cells that are protection are actually becoming overactive and harming us. So what we look at is there anything here that might be causing excess inflammation? Then we start looking at antioxidants. One of your favorite subjects, NRF2. So when we look at the genomics, we can see if NRF2 is underactive. The other thing we're going to talk about tonight is NAD, NADPH. We'll talk about how that is so important in so many functions. Well, that'll be most of the things we're going to talk about. Then glutathione, the master antioxidant, which is NADPH dependent. And then, just as important as glutathione, we have superoxide dismutase and catalase. And if we have a little opportunity, we'll discuss the sirtuins. They're quite fascinating as well because they control the production of SOD and catalase. They balance mTOR and autophagy, and we can talk about that just a little bit. mTOR is the growth of new cells. mTOR, without mTOR, the sperm and the egg never becomes the baby never becomes the adult. But excessive mTOR will feed excessive cellular growth. The inverse of that is autophagy, the cleaning of the cells. And I believe there's many genetic and epigenetic factors today that is upregulating mTOR and weakening autophagy. FOXO is another set of longevity genes that are related to the production of SOD and catalase. And I think that's all we'll have time to talk about today. This next row is your phase 3 detox, autophagy, your 5 steps of phase 2, digestion of fats, neurotransmitters, mitochondrial function. Then we get into a little bit of CYP phase 1, nutrient metabolism, and methylation from a building standpoint. But for tonight, we just want to focus on down here because this is where I believe most of the dysfunction is that we're seeing today that leads to an increase in inflammation. So I think when we focus on, or any of these out of balance, is any one of these creating more inflammation, or is any of these not making enough antioxidants, that's when we get that imbalance. So that's why I call this the 3D chess game played underwater. Multiple factors. If people are looking for simple solutions, we don't have any of those. And that's why I think we need to move to that functional approach, because I think someday we're going to look at the pill for the ill as being okay, but insufficient for the complexities that we're now seeing. Mm-hmm. Because unfortunately, we're living in a world that we didn't live in 30 to 40 years ago. What particularly concerns me is the plastics. You know, these plastics are xenoestrogens. They're now in our water supply from our polyester clothing. The ocean is filled with plastics. You know, I'm 65 years old. And when I was 12, the girls I went to school with looked like little girls. And that's changing now dramatically. Testosterone levels are dropping. Sperm rates have dropped 53% since 1973. We're going to look back on this and someday, what were we thinking? We give our animals growth hormones. Well, they'll get fatter faster and we can make more money. But just think what we've done. Those growth hormones get passed on to us. They stimulate mTOR. They stimulate inflammation. You know, one of the things that's controversial, but I think we're going to find, and that's EMF exposure. We really don't know what the long-term effects is of 24/7 being exposed to TMF. There's lectures out there that are excellent by Dr. Mercola and Martin Paul that talk about how the EMF may stimulate more calcium going into the cell, increasing the superoxide and the ionos to make peroxynitrite, a very nasty free radical. Then we genetically modify our foods. And, you know, there's still some disagreement on glyphosate, but it's certainly changing now. It's becoming more and more evident that glyphosate is having an impact on us. If we have time, we'll look at how it impacts potentially the heme cycle and sulfation. And then, unfortunately, some of us have learned about mutations like MTHFR and inadvertently, I believe, take too much methylfolate, which can then drive inflammation. So again, I believe we have to get away from this pill for the ill. Oh, I have MTHFR, therefore I need methylfolate. Maybe. And so that's the backdrop of what I believe is happening to us. potentially the reason why we're seeing such a dramatic increase in chronic illness among children. I mean, when you look at the statistics, chronic illness among children is skyrocketing. We used to see, autism used to be 1 out of 1,000, now 1 out of 110 or something like that. We're seeing inflammatory bowel disease among children going up. When I talk to teachers who are in elementary school who have taught more than 5 years, I'll ask them, how are the children today versus 5 years ago? Always. They say they're more anxious, they don't behave, they have a hard time focusing. I talk to college professors. Tell me about the freshmen, particularly the boys. Oh my, they are anxious. They need safe spaces. They get offended so easily. They're not rough and tough like they were. They're very fragile. So there's something going on, I think, in humanity. And even when you look at the recent events of how angry everyone is, I believe there's certainly changes going on from environmental factors. And those who might have some genetic weakness are the proverbial canary in the mine who get impacted more than others. My grandfather was a coal miner, and he told me about how they would take the canary with them. And when the canary fell over dead, they knew they better hightail it out of there because they were next. And I think some of the people that are so chemically sensitive and so ill today are the proverbial canaries. And they have genetic patterns that 75 years ago was fine, didn't matter, because we weren't exposed to what we're exposed to today. But throw those people with some genetic weakness in the world that we're living in today, and they're not doing very well. And it looks as though we have a new panelist there who's going to be helping us with.
Yeah, she's always does.
Yeah, and that's fine. That's cute that your kitty cat's there with you. So that's the background as to what we're thinking about. That we have to find that balance and make sure that we don't have excess production of free radicals, and we have adequate antioxidant protection. So any questions or thoughts or comments before we start moving into
Incredible.
NADPH? Okay, all right. Let me just highlight just a little bit some of the things that can cause this inflammation. Then we'll get into the to the other factors here as well. So let me just clear the screen, get the mouse back. So what I'd like to share with you, this is one of my favorite charts of what can cause excess inflammation. And this is called the Fenton reaction, discovered by Dr. Fenton in 1895, all the way back then. And what he described was that, inside the mitochondria, okay, once again, we make energy, and sometimes a free radical will fly off combined with oxygen to make superoxide. Then what can happen is we need superoxide dismutase to turn that into hydrogen peroxide. Then we need catalase, glutathione, and thioredoxin to turn it into water. That's provided that glutathione's adequate, thioredoxin's adequate, and your and my both favorite subject, NADPH, is adequate, because NADPH, as we'll learn later, takes your oxidized glutathione and whips it back into your reduced. If anything happens here, and we might have some genomic issues that would cause us to overabsorb iron, we make some nasty hydroxyl radicals. And then they combine with nitric oxide to make, oh no, peroxynitrite, one nasty free radical, which, by the way, depletes your glutathione.
Nasty.
So the more peroxynitrite you have, the more your glutathione is depleted. So there's the one way that we can make some extra free radicals. Then I won't show all of these, but I just want to very quickly illustrate. This is one of the other ones that I think can really make a lot of free radicals, and that is when we have what's called NOS uncoupling. So there's a molecule called BH4, tetrahydrobiopterin, and it needs to combine with, again, our favorite subject, NADPH, and oxygen, and arginine by the NOS enzyme to make nitric oxide. Now, most people know nitric oxide, Nobel Prize, 1980s, on dilation of blood vessels, but it also calms down mast cells. That's ideally. If something happens that we don't have enough BH4, or we don't keep recycling BH4 back and forth to BH2 adequately, we're BH2 dominant, or if we don't have enough NADPH, rather than making nitric oxide, we make superoxide. That's not bad enough. That combines with the nitric oxide we do have. That makes more peroxynitrite, further depleting our BH4, and we have one nasty little pattern that just keeps going here in NOS uncoupling. Okay, so, and you can see here again, this is NADPH dependent. So we really need our NADPH in so many factors, and I believe I'm going to be making an argument tonight when we talk about the NADPH steal that once again, due to epigenetic factors, amplified in those that are genetically weakest, our NADPH is being depleted. So those who have less than optimal production of NADPH combined with someone who may have genetic and epigenetic factors that uses up to make inflammation are the ones that are really struggling. And in our practice, this is what we see happening. Those who might have genetic predispositions to less than optimal NADPH production that we'll talk about, and then it gets used up by the NOx enzyme. These are the folks that have so much inflammation going on. They've been to 15 clinics. Nobody can figure out what's going on because traditional things to knock down inflammation are ineffective, and we're going to try to cover that here this evening. So there's the two major areas that I believe we make excess inflammation. So now what I'd like to do is I'd like to dig in a little bit to glutathione. And I'm sure everyone who's on this call, knows the benefits of glutathione. Master antioxidant through glutathione peroxidase takes out hydrogen peroxide that we just showed can create the Fenton reaction. And then through glutathione conjugation, your phase 2 detox that takes out a lot of your toxins. Of course, we start up here with cytochrome P450. Then we make our xenobiotics, and then phase 2 comes along, and then, we excrete it in the stool or the urine. Well, that's not going to happen if we don't have enough glutathione. And there's a lot of things genetically that can happen that can impede your body's ability to make the glutathione. So here you'll see on this chart, you can see that your homocysteine comes down from your methylation cycle. Here's your cysteine, turns into Y-glu-cis, glycine, and glutamate, and the three of those combine by the GSS enzyme to make reduced glutathione. Then the GLRX enzyme is what takes that reduced to help it through glutathione conjugation. Now, once it does that, this is where it gets really tricky. That becomes oxidized. So this reduced glutathione does its job and becomes oxidized. And we need NADPH and the GSR enzyme to take this guy back up here. So if we don't have enough NADPH, GSR is not working, we get stuck down here in oxidized glutathione. Now, it just doesn't sit here. It combines with oxygen to make superoxide. Then it combines once again with nitric oxide, chewing that up. Here we go, peroxynitrite, a very oxidizing agent, which then chews up your glutathione and also combines with CO2 to make carbonate radical CO3, which actually damages your DNA. So many times, well-meaning physicians have seen someone who's inflamed and they say, okay, I got you covered. I'm going to give you glutathione or a Myers cocktail. And of course, many people it helps. Some people feel worse. So the question is, why do you feel worse? Well, if you put that glutathione in and it does its job and it doesn't recycle back up, we're going to go down this pathway. And that is what's so confounding. We tend to think glutathione, the master antioxidant, the lower it goes, the sooner you die from all causes. We got to have it. But in some instances, taking it can actually make you worse. Hence the 3D chess game played underwater. Very complex. So we're going to be looking at all the ways that we need to make NADPH, but we're also going to look at how this NADPH might be used up excessively. Okay. Now, your other favorite subject, of course, is NRF2. And, you know, we could do hours of lecture on NRF2, but we're going to make it incredibly simple. And that is that NRF2 is what stimulates, you can see these arrows here, the production of glutathione, the utilization of glutathione, the recycling of glutathione. So I give it a simple analogy when, you know, working with clients. I say, think of a sprinkler in the ceiling, and it sits there with water behind it, and year after year, it can do absolutely nothing. But if a fire starts, keep one releases NRF2 and says, make some antioxidants, use them, and do your job. And in our assessment, we look at the genetics of NRF2, and if there's mutations on NRF2 or KEAP1, well, that may not be as robust as we'd like it to be. So again, I give the simple analogy. The fire can start in the body, and the body says, what fire? I don't see a fire. You see a fire? And it does not respond and turn on all these antioxidants. So what an absolute miracle we are that, you know, we have this sensing device that senses inflammation and releases this to put out the fire. So if we don't have adequately working NRF2, or we have mutations in it, we do not respond to that inflammation. And just observing over the years, we have seen that when people have a lot of mutations on NRF2 or KEAP1, these are folks that have a lot of inflammation and no one's able to seem to help them because they're just not responding when inflammation comes along.
Let me just interrupt you for a second, because obviously as a breast pathologist, I'm seeing something that is very attractive to me, aka BRCA1. And the interplay between the BRCA1 and NRF2, the crosstalk and stuff.
Yes, and you know, that's controversial. You know, some people, you know, see that they have BRCA1 and they get their breast removed. And again, I am not a geneticist, but here's the Bob Miller opinion. I think that, you know, if you look at people who get breast cancer, many of them have mutations on BRCA1, but I would supposition there's probably a lot of people that have mutations on BRCA1 and never get breast cancer. Because I think it's so multi-dimensional. So if BRCA1 impacts your NRF2, that may be significant if you have a lot of toxins, if you have some other mutations down here, you know, if you've been exposed to things, if you're under a lot of stress. I tend to think if all you have is BRCA1 and everything else is okay, and you're not exposed to a lot of chemicals and you eat well, you may go a whole lifetime without getting breast cancer. So I think we're, you know, I think that's where we make a mistake. And same way with the APOE. Is that a factor for dementia? Sure, but I have one of the APOEs and I'm almost 66 and I think my brain's doing okay. Now, I got to work on it, but I have to do a lot of things to keep the inflammation down. So I don't think these mutations are death sentences. And I think we scare people by telling them those things. And again, 3D chess game, BRCA plus weakness, you know, plus maybe the Fenton reaction going on, NOS uncoupling going on, living in a house with mold, living under incredible stress. Well, that might be a prescription for breast cancer. Yeah. So there's the story on NRF2. Now let's move over to one of my favorite subjects, and that is NAD. And by the way, this, if any doctors would like this map, we can provide it to them. This is the map that I made. That's the schematic that I call the inflammation pathway. I have it up on my wall, and I have it. And in our software, we're now getting to the point that that we can actually have this in the software lighting up with the genetics of the individual that we're looking at. So we're trying to be on the cutting edge. But for right now, let's look at NAD. So you'll hear a lot of lectures, talking about the importance of NAD. Clearly, everything they tell you is correct, okay? So what happens is, and I'm going to go through this, there's a process where we make this NAD plus, and then it goes a couple of different directions. It will move up to NADH, and again, here's the IDH3. So if someone has a lot of mutations on IDH3, this may not happen. Of course, we all know that NADH is at the top of the electron transport chain, and this is where we make our energy. But one of my favorite enzymes that I really keep an eye on is NQO1. And I believe in that's, people tell me what are one of the top 10 mutations that you're concerned about. I would have to say it's the NQO1, because this is NADH to NAD plus. And the reason I believe that's so important is if we're stuck up here, Bob Miller observing, clinical observation, nothing else. These are the people that many times have seizures, migraine, headaches, tremor type of conditions, because they're not coming back down. Now, why is it so important to come back down? PARP is what uses DNA, but it's our DNA repair. So when our cells get damaged, the repair team comes in, the construction crew comes in and says, let me fix this up. If you remember, when we make peroxynitrite, that damages the DNA. PARP tries to repair, but it's NAD plus dependent. So if we don't have enough NAD plus and our cells get damaged by peroxynitrite or radiation or just aging, we do not have repair capacity. And I think that's why you can look at two people that are maybe 50 years old. One looks 35 to 40 and another one looks 70, because they've not have the ability to do that DNA repair. That's all NAD plus dependent. Then through the NAD kinase and IDH2, we make NADPH, my favorite subject. Okay, and I'm going to come back to the NADPH. But let's look what else NAD does. It supports the sirtuins, which look what they do. They turn glutamate to alpha-ketoglutarate inside the mitochondria. So we're not going to have very good mitochondrial function if we don't have SIRT3. But SIRT3 is dependent upon NAD+. Inside the kidneys, we have the urea cycle. This is where we take ammonia, turn it into urea. If we don't have proper SIRT3, this may be less than optimal. Then we have FOXO3, which stimulates catalase and superoxide dismutase, incredibly important antioxidants. Then SIRT1 is needed for your vitamin D receptors. So I'm sure, as a physician, you've seen this. People with low vitamin D, you give them vitamin D, and the vitamin D doesn't go up. And that, there's many factors, but one of them can be lack of SIRT1 activity due to mutations or not enough NAD+. And then the same thing here, it involves the FOXOs and the SOD. Now, I'm a huge fan of mTOR and autophagy. mTOR is the growth of new cells. Autophagy is the cleaning of the cells. And if we don't have mTOR, the sperm and the egg never becomes the baby never becomes the adult. If we don't have autophagy, we don't clear out the toxins. And a good sign that autophagy is not working is you start to get a lot of age spots prematurely. So autophagy takes out the dead cell. So guess what? SIRT1 inhibits mTOR. SIRT3 supports autophagy. Now, what's interesting is that the COVID virus hijacks mTOR for replication, and autophagy helps take it out. Now, you can have mutations in ULK1, ULK2, and ATG13 that we all look at in our software reports that will stimulate autophagy. I did a study on Lyme disease, and I think it was 2018 that I presented in Boston. And we spoke about how those with chronic Lyme seem to have mutations that would allow epigenetic factors to throw off that balance. And this is the chart that we actually used and presented as the poster presentation. mTOR, the growth of new cells, autophagy, the cleaning, and mTOR will stimulate inflammation. Xenoestrogens that we talked about, the hormones,
Yeah.
EMF, all of those things are pushing our mTOR.
Yeah.
And I believe this is why we're seeing some of our health challenges today. And this is also why there's such an interest in intermittent fasting and ketogenic diet. Because when we intermittent fast, we take away our amino acids and carbohydrates and folates. Think of mTOR as the construction crew. Think of this as the janitors. When you take away the fuel, the supervisor says, hey guys, I don't have any material, let's stop building. Okay, janitors, come out and do your thing. And I believe genetic and epigenetic factors are throwing off this balance. So when people do intermittent fasting, where they eat for 8 hours and fast for 16, or go ketogenic, what they're really doing is they're supporting their autophagy. And that's why I believe some of the people that are at most risk for some of the illnesses we're seeing today are bodybuilders, because they do the growth hormones, they do the amino acids that stimulate the mTOR, and they might be building muscle, but they are putting this mTOR autophagy balance way off, putting themselves, I believe, at serious risk. So that's why that NAD is so important, because if we don't have NAD, we don't get our antioxidants, we don't repair our cells, and we don't have balance between mTOR and autophagy. Now, as we said, it also comes down here and makes NADPH. Now, why is that important? So I looked over here and I put all the things that NADPH does. So NADPH, as we said, is a cofactor for the NOS enzyme. So if everything is lined up, but we don't have enough NADPH, rather than making nitric oxide, we make superoxide. Most people know what that does now, combines with the nitric oxide you do have to make peroxynitrite. Okay. We spoke about this briefly, but when I showed you the iron, we need an antioxidant called thiodoxin. A lot of people are not familiar with thiodoxin, but it's a very important antioxidant. And once again, we need NADPH to take it from oxidized to reduced. We talked about this with glutathione. We need NADPH to go from oxidized to reduced. Now, when we make something called heme, when it's aged and it's ready to be discarded, NADPH and cytochrome P450 put that heme into ferritin, which is storage. So a lot of people are low in ferritin. So what do we do sometimes? Well, we give them more iron. Well, that's okay sometimes. But if this is not working and the iron is becoming dumped, you can actually make the situation worse by giving more iron. So I think it's important and incumbent upon us to know, is the ferritin low because of a bleeding disorder or not enough iron, or not enough NADPH, and that the enzyme here is heme oxygenase, which I'm very fascinated by. We're presenting at a NILADS conference in a month on the relationship of heme oxygenase and COVID, going into the cytokine storm, not whether you get it or not, but whether it goes into the cytokine storm. Heme goes into biliverdin, which is part of the antioxidant that neutralizes mast cells. And it also makes the CO that stimulates NRF2. Oh, yes. Yeah, the 3D chess game. That's possibly why, and again, now I'm off to speculating. That's possibly why some breathing techniques where you hold your breath for a little bit may be actually stimulating NRF2. Can't say for sure.
Yoga.
Yeah. Yeah, can't say for sure. Now, here's where it really gets interesting with NADPH. And the reason I'm so fascinated by NADPH is that it wears two hats. The one hat that it wears is doing all of these good things. However, what a miraculous organism we are. And when we are faced with a pathogen of some kind, a bacteria, parasite, something else, the body says, red alert, we got a problem here. We need to create some free radicals. Now, we tend to think of free radicals as bad. In excess, they are. But without them, life wouldn't exist. They act as signaling molecules. They also kill the pathogens. So when a pathogen comes in, the body says, iron, give me some oxygen. NADPH, give me an electron. Let's create some superoxide. Let's create some hydrogen peroxide. Let's activate our mast cells. These are white blood cells that are like the soldiers or the police officers. They're floating around just waiting for a problem. And when they're called upon, they create cytokines. They create histamine. They kick up quite the fuss, and they make all kinds of inflammation to kill the pathogen. In studies where this NOx enzyme is taken out, the animal dies of infection. We need the NOx enzyme to make inflammation and mast cells. However, there's a balance to everything. And one of the things that we've been researching is how epigenetic factors, things in our environment, are upregulating this NOx enzyme. And when these mast cells are excessive, this is where they cause excess histamine. And as you know, we need cortisol from the adrenals to knock down the histamine. How many people in your practice do you think have adrenal fatigue?
A lot.
A lot, yes. One of my favorite jokes is, how do you know if you have adrenal fatigue? And the answer is, if you live in America. So when we make all these mast cells, we make all this histamine, we can deplete our cortisol. As a side note, cortisol comes from progesterone, and there's actually genetic mutations in one of the CYPs that inhibits the progesterone to cortisol conversion. So interestingly, whenever we see that, we'll do like a forced saliva sample, and many times these people are low on cortisol. And I'm sure all the physicians who are watching this know that cortisol turns into cortisone, which is anti-inflammatory. That's why we give prednisone and other corticosteroids. So as you can see here, the hypothalamus controls all this down to the adrenals. And interestingly, if you listen to any of the lectures from Dr. Theo Hardy's from Tufts University, who's an expert on autism, he believes that mast cells are stimulating the hypothalamus, causing many of the emotional conditions we see in autism. So here's where it gets very interesting. NADPH wears two hats. It will do all of these good things over here. However, when the NOx enzyme sees danger, it says, oops, gotta go. I gotta go over here and I have to make some inflammation. Now, at first I thought, why would something that does all this anti-inflammatory stuff be inflammatory? And then it came to me that, well, what's likely happening is that when the body has a foreign invader coming in and needs to create oxidative stress, it wants to temporarily...
I'm sorry. So sorry.
No worries.
So sorry, I'm back.
Okay, it wants to temporarily shut down our antioxidants so we can make an inflammatory burst. Again, this inflammatory burst isn't bad if we are fighting a pathogen. Without it, we die of infection. But when it's running all the time, this potentially could be why we're seeing a rise in autoimmune disease. Now, that's purely speculative, but there's a lot of doctors thinking along that line. So let's look at some of the things that can cause this. EMF, sulfites. Now let's briefly talk about sulfation. Sulfation is where the body takes sulfur, combines with the salt enzyme to take out catecholamines, hormones, xenobiotics, and toxins. Suox enzyme is what turns sulfites into sulfates. Fascinatingly, if this doesn't work, the sulfites stimulate the NOx enzyme, and what is not sulfated, like dopamine, stimulates the NOx enzyme. And just turn on the news for 10 minutes, and I think you can get a pretty good feel that dopamine might be just a little bit high these days because everyone's so on edge. Now, why might that be happening? This is controversial, but if you listen to the lectures from Stephanie Seneff, PhD from MIT, she believes that glyphosate is mimicking glycine, which is what combines with succinyl-CoA from the Krebs cycle to go through the multiple steps of the heme cycle. So these enzymes right here all take that succinyl-CoA and turn it into heme. Now, again, we tend to think of heme as hemoglobin, but look at all these other roles that it takes. Neuroglobin, cytochrome P450. Your phase 1 detox pathways are dependent upon heme. Your electron transport, your peroxidase, your catalase, your NADPH, your tryptophan, your nitric oxide, and SUOX are all heme dependent. So if glyphosate, and that's an if, because it's debated, but if glyphosate is impacting this, it's impacting this pathway. Now, what's also interesting, this is not debated, and that is that these porphyrins that need to be turned one into another, okay, if they are not transported one into another, here's what happens. There's a neurotransmitter called glutamate that makes you intelligent, highly motivated go-getter, turns into GABA, but those porphyrins will block the GABA receptor sites, leading to anxiety. And how many of your patients are dealing with anxiety? Probably a lot. So and I remember talking to pediatricians saying, they used to deal with ear infections. Now they feel like they're dealing more with emotional issues of anxiety among children. So those porphyrins will block your GABA receptor sites. As a side note, inflammation will also block glutamate to GABA. So, and then interestingly, glutamate also stimulates the NOx enzyme. So this circular pattern just keeps going on. Now, one of the things that we've really been digging into recently is the RAAS pathway. As we know, there's a molecule called renin that stimulates angiotensin 1, then angiotensin 2, and aldosterone. And as we all remember, aldosterone causes you to hold on to sodium, excrete potassium. And of course, that will cause high blood pressure, and that's why physicians will give ACE inhibitors like lisinopril. However, there's an enzyme called ACE2 that turns angiotensin 1 and angiotensin 2 into angiotensin 1-7. COVID, by the way, comes in using ACE2. But if you've got mutations here, this angiotensin 1 and 2 to angiotensin 1-7 is not as robust. What does aldosterone do? It stimulates the NOx enzyme. Oops. And then, as we know with the cytokine storm, we're talking about stimulation of interleukin 6. So angiotensin 2 stimulates interleukin 6. Interleukin 6 stimulates the NOx enzyme. The NOx enzyme stimulates interleukin 6. Another feedback loop. Now, if that isn't exciting enough, okay, this is what we've now looked, again, we dug into the literature on this. This is all, peer-reviewed literature. Here's what we spoke about, but when the NOx enzyme is upregulated and it makes superoxide, as we said, it combines with nitric oxide to make peroxynitrite, stimulates the mast cells, stimulates the histamine. So we started, our research team started going through the literature, and were we ever stunned to find that peroxynitrite, mast cells, histamine, dopamine, and testosterone stimulates renin? Oops.
Oops.
Yeah. So what we're seeing is a negative feedback or positive feedback loop here, where once this gets kicked into gear, it just keeps going. And we've only, made this, again, not a discovery, it's all out there in the literature, but we just kind of connected the dots. All the literature is out there. We didn't, of course, do the research on this, but we did literature review, and by golly, and that's why, possibly why, high glucose is the other issue. This is possibly why diabetes, being a male or even being a bald male, is a risk factor for cytokine storm in COVID, because it stimulates the NOx2. A paper just came out July 25th that implicated NOx2 activation in the cytokine storm. So, and that's why possibly, some people didn't even know they had COVID. Other people are like, yeah, I had the flu, and other people are gasping for breath or dying because this is being upregulated. There could be many other factors, but this could be one of the factors involved in the interleukin 6. So I've called this the Holmes cycle after a relative of mine who's rather influential in my life. So I gave it, I gave this name the NADPH steal, where NADPH is used excessively, and then it just creates this cycle of inflammation. Possibly why, when we're exposed to mold and or Lyme, this thing just kicks off. You might get rid of the mold, you might get rid of the Lyme, but the pattern might be continuing to move on. Now, I'd like to wrap up in our last minutes here with talking about what can go wrong with NADPH. Again, there can be genetic mutations that can impair our production. So let's look at the pathway of NADPH. So there's an enzyme called G6PD, extremely common among people from Northern Africa, Southern Italy, some of the Bahamas and South America, and also Native American Indians. Now, what's interesting, by natural selection, those with G6PD were more protected from malaria. So those G6PD mutations, and I honestly don't know the mechanism, how it protects you from malaria, I'm not sure. But it somehow protects you from malaria. But back then, we did not have all the inflammation that was occurring. Now, what's interesting is Ashkenazi Jews, people from England and Ireland and some parts of Germany, by natural selection, have genetics in the HFE gene that causes them to absorb more iron. And again, during times of famine, that was a good thing because you absorb more iron. So the women who had those mutations were more likely to have healthy babies or have babies because they overabsorbed iron. So what Bob Miller has been clinically observing, that's it. Nothing more than just sitting in my office talking to people. Those who have English, Irish descent and they overabsorb the iron, and then Southern Italian or African together leads to the overproduction of the hydroxyl radicals and the underproduction of the NADPH. And I'm seeing the people who have the worst of both worlds, more production of free radicals, less production of NADPH, having trouble. Now, there's another pathway here where tryptophan, and of course, turkey, that's where we, it's an amino acid. It goes down this pathway to make serotonin and melatonin, but also goes down another pathway called de novo pathway. You can have genetic mutations in KYNU, QPRT. Any mutations along here are a problem. And interestingly, when we get stuck here, many of the people have lipid peroxidation and anxiety because that quinolinic acid is very excitatory to the brain. And then there's also ME1 that in the Krebs cycle inside the mitochondria makes NADPH, and another one in the Krebs cycle called IDH that makes your NADPH, and then that's right before your alpha-ketoglutarate in the Krebs cycle. So mutations here could lead to less than optimal energy production and less than optimal NADPH. So now you finally get down to here, and this is where when people take niacin, and nicotinamide riboside, nicotinamide mononucleotide is becoming very popular. People are taking a lot of it now because they hear about all the benefits of NAD+. Everything you heard is true. However, well, as we age, this also weakens. So this is why, as we age, if we don't make enough NAD plus, and we have more damaged cells, there's this tug of war between, do we use our NAD plus to repair cells or support SIRT? And we have some charts on that I don't have time to show tonight, but there's that tug of war between those two. So this is why people will take nicotinamide riboside or nicotinamide mononucleotide. I personally like the nicotinamide mononucleotide. Oh, and I forgot to mention on NQ01, Podiarco helps that beautifully. So one of the things I like to do to support NAD is grapeseed extract to support QPRT, nicotinamide mononucleotide, and Podiarco, those three together. So again, we tend to think, woohoo, take NMN. And, there are doctors who do nicotinamide riboside infusions. And I've talked to many of them and I said, so tell me, do you have people that this is like the miracle that's helped them more than anything else? Yep. Do you have some people that it was like they had a horrible reaction and it was like the worst thing they've ever done? Yep. So I'm now going off to the Bob Miller theory again. So not peer-reviewed, not studied. I'm just passing these out as hypothetical. But my hypothetical thought is that if the NOx enzyme is a runaway freight train and you push more of this, very well may be feeding this.
Here's an NAD still, NADPH still.
Yes. So I tend to think, just to be on the safe side, to be conservative, before we start pushing a lot of NAD plus, because we get on bandwagons. First it was MTHFR, we need methylfolate. Now it's NAD, and I think there's no magic bullet that fixes everything all the time. Life's not that simple. So I tend to think we have to make sure that we've got this guy calmed down, that it's only acting responsibly and when needed, not by aldosterone being excessive, not by excess glutamate, not by oxalates. We didn't have time to talk about that, but when we get leaky gut, oxalates come in, upregulated mTOR, sulfites, dopamine, homocysteine. There's a book out there that talks about the higher the homocysteine, the sooner you die from all causes.
That's right, including cardiovascular disease.
Yes, well, it's stimulating the NOx enzyme. So I tend to think what we need to do is we need to make sure this is not upregulated before we start aggressively pushing.
But I see you have a curcumin and spirulina there.
Oh yes, that calms down NOx, but getting rid of oxalates, reducing histamine, balancing mTOR, making sulfation work, making sure your dopamine turns into norepinephrine, you're methylating properly to get your homocysteine down, you're not smoking, your glutamate's controlled, your aldosterone is not too high, which is also stimulated by stress, mind-body connection. Make sure this is calmed down before you aggressively push your NADPH. That's what I've been recommending to the doctors that I train. So I think what we did there in an hour, we ended right on the hour. We tied together, the importance of glutathione, why we need it, but why we need NRF2 to support it, why we need NAD plus, why we need NADPH, but how we have to be careful that NADPH is not used inappropriately by the NOx enzyme to stimulate excess mast cells, excess histamine, excess glutamate that begins that home cycle that just spins around and around. So I think you were spot on in putting your attention to those molecules, NRF2, NAD, NADPH, because they are a symphony that plays a very complex orchestra.
This is so incredible. And guys, now you probably do understand why was I so excited. My glutamate was so high because I had Dr. Miller talking, and I also warned you that that's not going to be a walk in the park. And he made it obviously, however, the comments are coming through that this absolutely amazing. This is, what you did, Bob, there is so much sense. Whether it's a Bob Miller's theory via peer-reviewed and the studies that you're conducting. But I think that Bob Miller's theory is absolutely spot on. And again, the bottom line is there's no one pill for an ill. There's no one pill for an ill. It is so multifactorial. But health begins with health. So guys, I hope you understood that glyphosate is no good. And glyphosate is your Roundup. That, yeah, we need to minimize our digital devices. We need to clean the lifestyle. Obviously, we kind of have to clean the stress and the food and all these components. And then we will start adjusting the symphony. We'll start playing the right notes.
Absolutely. Yes. Well, thank you for that. And if you don't mind, if I can just give just a little commercial.
Please, please do.
Yes. So
genetics doesn't pull the gun. I mean, epigenetics pull the trigger.
yes. We are going to be having a conference on mycotoxins.
Yeah, please do. Yeah, I know. Stephanie Seneff, guys, come on. It's on glyphosate, it's on sulfates, and it's on Alzheimer's. Hello.
Yes.
Dr. Jill Carnahan. Jill is amazing on GI, on microgut, on microbiome, on LPS, meaning the toxic stimulation of everything which comes to the brain. Like, hello.
Yes. So September 18 to 20, only $385, and just go to nutrigenicresearch.org. You don't have to put this in. You can get there. And 3 days. I mean, we're going to be spending 3 days on mycotoxins. And I do believe that mycotoxins are more serious than we ever realized. And they do stimulate, if you go back to the map, the mycotoxins right here are stimulating the mast cells. So I think it's way underappreciated. So it's hard to believe, but I had a hard time getting the schedule in. We're going to do 8 or 9 hours a day on mycotoxins, and it is being.
Maybe that is exactly why I ain't going to register. No, I will, because it's like I'm not doing virtual 9 hours a day. I will.
Yes, well, it's all recorded, so you can watch it later. Yeah, I'm. When I do my seminars, we're brutal. We when we were in Denver last year, we went from 8 a.m. till 7 p.m. every day, with just short breaks. So when you go to my conferences, we're not there to party. We're there to learn. So.
Absolutely, guys. Take a look and stop. Yeah, obviously, 385 registration. When you go to some of you, when you go to medical conferences, what's the registration fees at?
Sure, and there are no CEUs, and or CMEs rather, and because we do want to talk about things that CMEs would limit us, but it is being recorded. We recognized we made a mistake. We put it on a Jewish holiday, but we won't do that.
Yeah.
But it is being recorded, and it's going to be on a service called Spot Me that's just top rate, and should be easy to navigate, and they get all the recordings. So I hope we covered all the subjects that you wanted.
Oh, that was incredible. The comments are there. It was incredible. I'm so excited to get the recording, because this is not a one spot. This is not one type meal. You kind of need to revisit and revisit and revisit. But then when it makes sense, then you start understanding health.
Yes. Well, thank you for this opportunity. If you'd ever like me to come back.
Bob. Thank you, Dr. Miller. And hopefully
Bob.
this is not the last one.
Oh, no, we'll do more. We'll do more.
Awesome. Guys, thank you. Thank you, everyone, for attending. You see, excellent. And guys, I will post the recording, most definitely. So thank you. We'll say goodbye and good night.
All right. Have a great night.
Ciao. Thank you.